I think throughout research, it is sometimes you have that disconnect where you're coming in every day to work, doing this experiment, and I'm going to get a result. I get a result or I don't get a result. But when you do see patients and parents of people with schizophrenia, it really puts things into perspective. It's also about making a meaningful difference.
The Hodge Centre for Neuropsychiatric Immunology focuses on the immune processes in mental health disorders, providing hope for patients suffering from some of the most debilitating mental conditions, such as schizophrenia, bipolar disorder, Alzheimer's and dementia. The Hodge Foundation have supported 7 PhD students starting their careers in this newly defined area. Today, we want to tell you about just one of these students. Today, we want to tell you Jack's story. Jack Redaway is a PhD student graduating in winter 2021 who has been supported by the Hodge Foundation for the last 4 years. Jack's research looks into how the complement system is associated with risk for psychiatric disorders, specifically schizophrenia. This link between the immune system and brain disorders offers a relatively new route to develop treatments for those at risk of developing these conditions. As part of his research into the immune cells in the brain, Jack developed a machine learning model to analyse larger datasets than ever before, giving more accurate results and a better understanding of what is happening in the brain.
So the reason I sort of came to both immunology and neuroscience was I had an interest in sort of both areas during my undergraduate. And then I had a placement year where I worked in a lab which had sort of a neuroimmunology bent to it, and sort of seeing how that was such a new frontier for science and sort of, well, in bioscience research, and sort of seeing how those two areas could be brought together where, it was the idea is we have these two huge bodies of information from, decades of research, but actually how the two interact is, completely new. So microglia are the immune cells of the brain. So they're doing the same job that other cells are doing everywhere else in the body, but they're in the brain. So they're specialized for the brain. And when they get stimulated by an immune signal, they change their shape. So they go from this sort of surveying shape where they're sort of constantly surveying their environment for danger, and then retract all their processes and change their shape, because this means they're now responding and trying to resolve that stimulus, whether it be a bacterial infection or damage because you've hit your head or something like that. So by looking at that morphology, we can see, well, okay, if they're looking like this, then the brain's healthy and fine. If they're looking like this, or more of them are looking like the activated morphology, then we know, well, something's going wrong in the brain, and that might reflect and tell us what's actually going on and what's happening, and also what processes are being engaged. The microglia prune and shape the neurons in the brain, which then control and affect behavioural outcomes. So whether it is in associative learning or other learning and memory processes, we know that if the microglia are activated, they might be performing these functions. So by understanding their morphology, we can then predict and say, look, the microglia are now functioning and they're the ones that are driving these behavioural processes. The idea is to understand the mechanisms behind how the microglia can do these, where they're doing it, what regions of the brain, whether it's the hippocampus, the amygdala, the prefrontal cortex. Understand where these events are happening, what time points they're happening, what stage of learning they're happening, and then also, if you do get an immune stimulus in early life, whether that's an environmental stressor or an infection, how does that then affect how learning occurs? So in order to look at morphology, we need to have images of the microglia and then have some readout of how they look. The standard procedure is to get a couple of images of your microglia, compare them across several groups depending on what your conditions are, and then look at how the morphology changes. So you might sometimes measure the length of a process, or you might measure the overall size, or you might just manually by eye go, that looks activated, that doesn't look activated. What my work's done is sort of do almost the same thing as that, but do it on a huge scale. So instead of gathering a couple of images, we've gathered thousands and thousands of images, and from those we get tens of thousands of cells. We look at their morphology, so we look at their process length, their surface area they cover, what they might be doing in terms of their shape, and get quantifiable data from that, but across thousands and thousands of cells, and also in a non-biased way. So everything else before is sort of done in a way where you might pick an easy one to trace, or you might want to pick ones that you can easily see and tell, and the ones in the middle you kind of give up and don't bother with. Because there's no human interaction here, we don't have that control. It just does everything according to the criteria we've set. And then we get a huge, huge amount of data, more than's been done before. And then we then analyse that in a way, again, that's non-biased with our machine learning model, where we use the machine learning to predict and tell us what's happening to that microglia, not based on human judgment. Talking about something the Hodge Centre's done is we sort of got to various stages. Initially we went into this not thinking we're going to use machine learning. We were like, oh yeah, we're going to get a load of data from this. And we kind of got to the end and went, right, we need to come up with a better way to analyse this data because the current methods we're employing aren't going to work. So that's when we sort of, within this research environment that we've got here, we went out to some of the bioinformaticians that were in the same building as us, and we spoke to them because of the collaborative environment that we have here. And they were the ones that were able to help us and push forward with this. And it's another example of how we wouldn't have been able to do some of the basic neuroscience without Jeremy. We wouldn't be able to understand the immunology and sort of the background to the microglial study we were doing without Paul. But then the analysis again was requiring this collaborative environment that we have here, and we were able to speak to groups outside of the Hodge Centre and actually work with them to sort of further our data as well. Coming to the end of my PhD and submitting it, it's been a long journey, and especially sort of with COVID and everything, it's sort of taken a bit longer than it was meant to originally. But it's nice to finally sort of be able to start showing people this work and sort of presenting it in a formal way rather than sort of just showing it informally to sort of colleagues and lab mates, and it's sort of now being able to go forward and sort of now take stock of what I've done, but also now plan the next experiments, where we're going to take this forward, what we're going to do next, and how other people might be able to use it, because we're now trying to package this in a way that we can share it with others and other people can benefit from what we've developed here. Because as we've touched on before, the idea is that it's not just in schizophrenia these microglial changes are happening. They're widely reported in traumatic brain injury, Alzheimer's, stroke models, depression, PTSD. So that's the stage we're at now, is now trying to develop this into sort of a tool that other researchers within Cardiff and outside of Cardiff can use. I think throughout research it is sometimes you have that disconnect where you're coming in every day to work and you have this, coming in, doing this experiment, and I'm going to get a result. I get a result or I don't get a result. But when you do see, we've had meetings where patients and parents of people with schizophrenia have come in and spoken or been in talks and things like that, and seeing, how just basic research and the fact that people are looking and are interested to understand why they or their family members are unwell or suffering with these symptoms really puts things into perspective. It's also about making a meaningful difference, and that's something that has to shape research as well. We have to be asking research questions which are translational and actually have the potential to make an improvement to people's lives.
At the moment, a lot of our research is really trying to understand the basic mechanisms of how the immune system affects the brain. Yeah. But we definitely see that it's not just defending against infection, it's really at a fundamental level affecting how the brain does some of its key things, like encode memories. And in some mental health conditions like schizophrenia, then we know that the brain processes information, including memories, kind of incorrectly, and that leads to some of the symptoms. In other conditions like Alzheimer's disease, sadly, memories are lost or harder to retrieve. And so if we can modulate those processes, that's really key to those kind of conditions. And in terms of both of those conditions, we see now that there's much more evidence that changes in the immune system in the brain are actually important in how they arise as clinical conditions. And therefore, it's a really exciting area in terms of targeting new treatments.
Jack has really brought a new avenue of research into the group. I'm an immunologist who's been interested in brain diseases for a long time, diseases like multiple sclerosis and Alzheimer's disease, diseases that we've always thought of as organic diseases. bringing Jack on board and working with others in the psychiatric space in diseases like schizophrenia, depression, etc., has really been a game changer for us and has taught us that there isn't really that much difference between those diseases we used to think of as organic and those we used to think of as psychiatric. They're really part of the same spectrum of brain diseases.
So Jack has been a great PhD student to have in the group. He came with an interest in how the immune system might affect the brain, and he's really run with that idea. He's particularly studied the complement system in the brain and has come up with some really exciting insights into how the complement system might affect memory. But he's also been able to develop new technical ideas and approaches which have really been able to take forward not only our work but also work in other groups in Cardiff and beyond to look en masse really at functional changes in immune systems in the brain.
I think the obviously big data is really something we in research are now having to manage all the time. We work with much larger sample sets. We often have to combine many thousands of samples collected across many different nations to get meaningful data. And I think good tools to enable that to happen smoothly and to happen in a way which is reproducible are really critical to us if we're going to get the best value out of that data that we're suddenly submerged with. So Jack's work has enabled us better to take very complex data sets and analyse and interpret the meaning of them.
There's two sides to this really. Firstly, as new fields open up, you need to be able to train the next generation of researchers who are going to learn the different skills that need to take that area forward. Many of the things that advance in science are at the frontiers between different areas, and in this case we're talking about the frontiers between immunology and neuroscience. And then you need to have people who can be trained in both those skills, and Jack has come and has learnt those different skills here in Cardiff. On the other side of the coin, you also need people who can bring new ideas and run with the ball and really take things forward into areas that they weren't in before. That requires different types of thinking and different approaches. And I hadn't suggested to Jack that he develop these new approaches. He thought of them himself and in collaboration and discussion with other colleagues, and was able to take that opportunity forward and really develop it. And you need that kind of fresh thinking to drive an area forward.
Oh, it's crucial. It's the most fun part of it anyway, because you get people who start off knowing almost nothing, coming out of a university first degree and having had very little experience, particularly of the practical aspects of research, and then over the course of 3 years watching them transform into independent scientists who are owning their own work and are able to deliver complex experiments in a way that they couldn't possibly have dreamed of just 3 years previously.
So neuroimmunology research really, I think, is amongst our best hope for understanding why many common neuropsychiatric conditions occur in so many people, and also to help mitigate the risks so that people can get faster and more accurate diagnoses and also more appropriate treatments to manage the severe conditions going forward. Our researchers have used the Hodge Foundation's investment to leverage hundreds of thousands of pounds in additional support from a range of different funding bodies who all recognise the crucial work that we're doing in the centre. Rather alarmingly, there have been very few breakthroughs or new treatments for mental health disorders in decades, and the treatments that we do have are really limited and often come with really unpleasant side effects. And I firmly believe that the Hodge Centre is now best placed to find new, novel treatments that will make a real tangible difference to patients. So today we've told you about Jack's journey and the difference that he, just one student, has made to this new and pioneering area of research. But Jack is one of 7 students that the Hodge Foundation has supported, and his successes can be mirrored across all of their work and their achievement. And because this area is so new and so unique to Cardiff, the training we've given to Jack and his PhD peers on the PhD programme is something I think that we can all be truly proud of. And we've created a cadre of researchers who'll go on to be research leaders in this new and I think very exciting research area. And I think that by equipping our specialist researchers, we've enabled new research, not only here in Cardiff, but also across the world, giving hope for patients struggling with these often extremely difficult diseases. So finally, at Cardiff, we are so grateful for you for supporting us and this pioneering research happening at the Hodge Centre for Neuropsychiatric Immunology and providing us with now what is a very solid platform on which to build opportunities for our research to progress into the future. Thank you so much for your support.
Thank you.
Thank you.
Thank you for your support.
Thank you.
Thank you.
Thank you for your support.
Thank you.
Thank you for your support.